So our next speaker is Amy Singh from ROW, which is a CRO or contract research organization. And again, I think this is probably a nice contrast. Kind of the giant kind of multi-year, multi-institution approaches versus like kind of similar to presentation, a little bit more adopting existing tools. And so her talk is entitled "Using Web Technologies to Improve Data Quality."
Thank you. So as you mentioned, I am from ROW. We are a CRO. We manage clinical trials. And half of the company does commercial work for big pharma, and the other half, which I work for, is the federal division. So currently we manage, in my consortium that I currently manage, we have 21 organ transplantation trials that are funded through NIAID. And we came to LabKey for a pretty specific need. So it's pretty much a contract from the multi-year applications that we've seen. So what the motivation between for us to go into LabKey is that with the 21 organ transplantation trials, they're enrolling between 200 to 500 participants, and they are 5-year observational trials looking for mechanistic biomarkers for rejection and various phenotypes. And we have 50 core labs, up to 52 now, core labs that we work with. And each core lab has multiple spreadsheets that they give us. And we manage PCRs, Luminex, Flow, so it's all coming in spreadsheets. So we were drowning in spreadsheets. So it was just a really messy process. They tried to do a custom program spreadsheet where they would pull all the clinical data from the clinical database that would already pre-fill the participant ID, the visit number, and collection dates in the spreadsheet before passing it on to the lab for them to return the results to us to help quality control. But that resulted in the lab having to manually copy and paste data blocks into that spreadsheet. And so that was one of our main selling points to the lab, that they don't have to do that anymore. So it was very well received because they were like, hey, we don't have to try to map our ELISA plate data to whatever spreadsheet it is. And so we are able to take their format. But then we wanted a real-time quality control reconciliation done in the system at the same time. So there was like I said, the spreadsheet, there was a lot of programming effort for the old spreadsheet, and there was no normal version control. It wasn't a secure transfer. They would just email it to us. And so we came up with a list of objectives that we needed. So, one of the reasons we're here, we chose LabKey because the development team is experienced in handling all the different assay types that we are looking to import. And we also do not take raw lab data. It's all cleaned and data that is imported into the system. So right now we use LabKey as a relational database. The specimen tracking data goes into the system, and the clinical data sets, specific data sets, go into the system, and also the assay data gets uploaded into the system. So the phase one, we wanted the lab data acquisition and quality assurance done. And we linked the data. So back up, we have a pretty sophisticated specimen tracking system. The tracking system knows what specimens expected at each visit for the participants, and it tracks from collection to shipping to receiving at the lab to the recording of the final disposition. And so we use Sample-Minded for that. And every night, Sample-Minded produces a data dump that dumps into LabKey. So our current status, the specimen tracking data is auto-loaded nightly. And we do have the custom lab import modules complete. The lab's upload format, they could either do a cumulative or incremental files depending on how they run their assays. The data is QC'd against sample-minded data in real time, and LabKey immediately reports discrepancies requiring resolution in listings and assay progress report. The specimen status is summarized, and the lab data is not published until all the queries and resolutions are done in the system. So in thinking about the system, we did not want to duplicate any other available data sharing analysis resources. We wanted it to be pretty specific and a value added to the labs. And so currently it has been pretty well received. In the second phase, which we are running more of an issue, is the data visualization piece. So we want the labs to easily see what data issues were in the specimen inventory, which we have. But then it's the actual assays, the outliers, visualization that we are having problems with determining. So it's finding the correct visualization. We have great programmers that can produce these fancy graphics, and then we have investigators who can't really tell us what they need. So we're trying to bridge that communication between the programmers and the investigators to get to that point. So let me show you the system via slides. This is our landing page. So this is, down the road we have more communication to the labs, and so this system is pretty heavily used by labs right now. And we haven't opened it up to investigators for visualization because we haven't gotten there yet. That's our second phase. So this was the dashboard. And here is what the specimen progress report dashboard looks like. So we have programmed different statuses. If there's a white circle if the specimen is inspected, and then if the specimen has been collected, the specimen has been received by the lab, and if their assay results available, or if there's a query. And in the system, there's also, we have customized the spreadsheet formats for them. So they would be able to download the spreadsheet format when they upload their process. And as long as the participant ID, visit ID, and date are in the correct format, the system will take the data they're loading. So once they populate their spreadsheet in the LabKey format, it gets uploaded into the system. And here's just an expanded view of what the assay progress report looks like. So with this is an example of a lab that we were having a lot of issues with. It wasn't the lab's issue, it was the collection site's issue. It's an ELISpot assay, and there were a whole bunch of cells that were not viable because it was received by the lab. And so it provided a great visualization for the investigators, and people would say, hey, you need to do something about your specimen collection. And we also had custom queries in the system that would total up where the status of the samples are. So it will tell us there's about 600 specimens collected, there were 6 not received, and it just goes down to how many final results we have for the assays. So here's the data format of what it looks like once it's imported into LabKey as the tabular format. So these are the queries that would fire. So in the progress map report, if there's a red triangle with an exclamation mark, there's an issue with that sample, they could go, the labs would actually drill down and see what the issues were. And if they uploaded two results, to look at the results for the same sample, it flags it and says, hey, you need to clean this up. There's 2 not matching results. And then there's others that they would, this one was flagged, the participant ID was not found, and that would be a site issue of not entering that participant ID in the specimen tracking system, so we'll have to go back to the site to have them clean it up. So the whole system is a lot of data cleaning and reconciliation efforts in trying to get the data in a system. Oops, what did I do? How do I get back? It's just another visualization of the three different specimen types that were from the same lab. So from one sample, we could actually split up to the three different markers that it was looking for and give a tally of what we have. Within the same sample, we might have different results for. Here's different in-system visualizations, and this is the step where we were having more of an issue getting to agreement of what is helpful for investigators. So we did form a working group with the main investigators, lab investigators that we have been working for in getting towards this. And the other reason why we used LabKey was because it is an open source and it does take R reports and JavaScripts and a lot of the current programs that the current programs are already using in-house, so they can just dump their visualizers and have a place to put them. And before this, they were creating all these fancy Java graphics, and they were all living in individual web-based programs. Like kind of have to poke around to see where you're at and where to find things. And now it's the place to house it all in one place. Another example. So the next steps, we are in the process of migrating all central lab activities in the active trials to LabKey. The progress has been a little slower than I had anticipated because these are 5-year observational trials and a lot of assays aren't done until the end of the trial because of batch effects. So there hasn't been a lot of labs that do ongoing assays, that they have assay results to load. So we're hoping as we're getting there, we'll get more labs using the system. And we need to complete the implementation. The specimen tracking data is auto-loaded, and that piece is worked out, but the implementation of the clinical analysis data sets, we need to work on that and figure out what is useful. The clinical database, of course, contains so many variables. It's kind of trying to figure out which ones are important and useful for the system. Like I said, we're forming working groups to develop guidelines and specifications for explanatory and end-of-study analysis, and the visualization features. And the new thing we were playing around with is this development of support for ancillary studies, permitting queries for samples based. So if we're looking for rejection samples across all 21 studies, we want to see if we could open it up so that the system would manage to be able to let us query for that. But I'd be interested in other previous predecessors and how their investigators felt about sharing that data. Because in our consortiums, the investigators, they're very protective of their own. So in how to get over that hurdle of opening that up and seeing where we go with that. So acknowledgements. The LabKey development team has been great to work with. All our custom development has been done by LabKey. We outsource the custom development with them, and I really appreciate Kristen and Matt and Avital. They are good at translating I'm not an IT person, and so they're great at translating what we need into specs and giving us what we need. And of course, our list of all the core labs that were guinea pigs and trying to get this data sorted out. Any questions?
So are you guys using this for people to be able to request the specimens? Is that the main driver, or just
We use it to organize, and it's a system to obtain the laboratory data. So we don't, for requests, we go through sample minded for that. So because they have a whole different system where they can ask for things.
So run those regulated trials, right?
Yes.
So are there any issues around the software and its level of certification?
Yes, so we have a very clear cut, and only specimen data, mechanistic assay data comes into LabKey. So all the clinical data comes through our electronic clinical databases, and those are all regulated and validated. Whereas because of these are biomarkers, and so it's all research purposes. So we make a very clear cut between the two.
Thank you, Amy. Great to see another sort of interesting use cases, and kind of to your questions, Tom, how different groups slice and partition data, between systems, and to be compliant with regulations, but still facilitate interaction.